Hello everyone. It is my pleasure today to introduce Dr. Michelle Poulin and Dr. Thiru Selvanathan of Fluidigm. Dr. Michelle Poulin earned her bachelor's degree from Smith College and her PhD in immunology from the University of Colorado at Denver. After completing a postdoctoral fellowship at National Jewish Health in 2003, she joined BD Biosciences as an instructor in the Customer Education Department before moving in 2012 to DVS Sciences as a field application scientist. She currently serves as the head of the North American Field Application Scientist team at Fluidigm. She is a highly experienced mass cytometrist and flow cytometrist, as well as an instructor skilled in the use and training of the CyTOF mass cytometer, multicolor analyzers, and high-speed cell sorters. Dr. Thiru Selvanathan earned her PhD in immunology from the University of Toronto, with a focus on invariant natural killer T-cell activation by bacterial products. For more than 2 years, she has worked as an application scientist at Fluidigm. She is involved in customer support and new product development, including the Maxpar Direct Immune Profiling Assay. She is experienced in the development of mass cytometry panels for immunophenotyping, and highly skilled in all aspects of experimental workflows underlying modern cytometry applications. We are excited to have Dr. Poulin and Dr. Selvanathan cooperatively present to us today a webinar titled High Dimensional Human Immune Profiling Simplified, 30 Markers, 1 Tube, 5-Minute Data Analysis. Thank you.
Today we will introduce the Maxpar Direct Immune Profiling System, a groundbreaking assay with a data analysis solution specifically designed for deep immune profiling of PBMC and whole blood samples in clinical and translational research. Fluidigm is well aware of the many analytic challenges faced when studying immune-mediated diseases and the complexity of immune response in health and disease. These challenges include limited and precious samples, longitudinal data comparisons, cost-effective and efficient analysis, incorporating new findings into ongoing studies, and the ability to find the unexpected. Flow cytometry is a typical way that immune profiling and monitoring is performed. Here is the 5-tube panel used by the Human Immunology Project Consortium, or HIPSI. While the HIPSI and other flow cytometric assays for immune phenotyping cover a number of markers, they require multiple tubes to do so, as shown here. Certain antibodies must be used multiple times across tubes in order to allow for deeper phenotyping of certain subsets. Correlative data for markers not repeated is lost. Thus, the number of populations that can be identified is reduced. Current fluorescence cytometry approaches to immune profiling have limitations that present challenges for studies requiring immune monitoring. These challenges include limited cell population identification, multi-tube labor-intensive assays, variability in site-to-site results, and subjectivity of manual gating strategies. When sample volume is limited or sample is precious, the ability to analyze all needed populations in a single tube is extremely valuable. Fluidigm has launched a groundbreaking approach to high-dimensional immune profiling with the Maxpar Direct Immune Profiling System, which addresses many of these challenges. The system consists of an assay kit with a dry 30-marker panel in a single tube, plus reagents needed to process either PBMC or whole blood samples. It also includes an assay-specific template to collect the samples on a Helios mass cytometer, and an automated data analysis software solution designed specifically for use with the assay. Data analysis of FCS files can be completed in as little as 5 minutes with Maxpar Pathsetter software. The Maxpar Direct Immune Profiling Assay with Maxpar Pathsetter software provides efficiency from a streamlined workflow. In the first step, PBMC or whole blood samples are stained in a ready-to-use assay tube. In the second step, data is acquired with a customized acquisition template on the Helios mass cytometer. In the third and final step, Maxpar Pathsetter software is used and population frequencies, data plots, and QC metrics are reported. Let's take some time to discuss the components of the system in more detail. First is the Helios mass cytometer. By enabling routine analysis of more than 40 parameters per cell, the Helios mass cytometer offers the most complete and comprehensive view of cell phenotype and function in a cytometric platform available today. The power of this technology allows you to collect the most actionable information from every cell in every precious sample. In addition, this is a proven technology and no longer considered to be a novelty only used by risk takers at a few institutions. It's now available at over 200 sites worldwide and is a key technology in multiple clinical trials. The use of fluorescence is still considered the cytometric standard. Although fluorochrome conjugated antibodies are widely used, they have limited utility for high parameter studies. As shown here, there is often significant signal overlap. Other challenges, such as highly variable fluorochrome intensities, background signal, and autofluorescence also exist. These challenges create limitations that impart significant complexities to experimental design and interpretation. Because of this, traditional fluorescence is currently limited in terms of routine use to 18 to 20 parameters at a maximum. Helios uses proven CyTOF technology, cytometry by time of flight, that surpasses the inherent limitations of fluorescence detection by using highly pure metal tags that are separated by mass instead of by wavelength. Metal tags have distinct signals which can be separated with 1 Dalton resolution, as well as less variable probe intensities. In addition, nothing analogous to autofluorescence exists when using CyTOF technology, since the metals we use as probes don't exist naturally in biological systems. These characteristics of metal probes provide a solution to the current challenges of high-parameter cytometry. Next, I'd like to introduce the second part of this system, the Maxpar Direct Immune Profiling Assay. The Maxpar Direct Immune Profiling Assay has multiple advantages. It is comprehensive, with the ability to profile 37 immune cell populations in a single tube with a dry 30 marker panel. The assay is also efficient and easy to use. The single tube with its optimized dry panel minimizes assay design and validation time while maximizing information for PBMC and whole blood samples. You just add your sample and go. And the Maxpar Direct Immune Profiling Assay is reliable. Its rigorous product QC and testing provides trusted results, lot to lot, run to run, and site to site. Here in this slide, you can see the 30 markers contained in the assay tube. The assay markers were selected with input from industry-leading pharma companies and academic institutions and builds on the panels recommended by HIPC. This assay uses the markers from the HIPC panel shown earlier in the presentation as its base. Markers colored in orange are those added, which expand the number of cell populations identified by the HIPC panel. These additional subpopulations include gamma delta T cells, neutrophils, eosinophils, and basophils, NK cells, both early and late, mate and NKT cells. These additions also allow for better definition of T cell subsets. CD45 was also included as a panleukocyte marker, and intercalator 103 rhodium is used for live dead discrimination. In this slide, you'll see the 37 populations identified by the Maxpar Direct Immune Profiling Assay for each sample. These 37 populations span the major immune cell populations found in PBMCs, lymphocytes, monocytes, and dendritic cells. When using whole blood, granulocyte populations can also be identified. The assay reagents are subjected to rigorous product QC, ensuring consistent lot-to-lot performance. Another way to look at the markers in this panel is to arrange them by the metals to which they are conjugated. Doing this enables you to see which metals or channels are available to add markers. In this way, the assay can be customized with markers of interest to you. When markers are added to the panel, the Maxpar Pathsetter software can be used to build a new automated analysis model. Now I'd like to introduce you to the automated software solution designed specifically to analyze FCS files collected using the Maxpar Direct Immune Profiling Assay, Maxpar Pathsetter. This software enables fast, reliable identification and enumeration of the 37 cell populations defined by the 30 markers in the Maxpar Direct Immune Profiling Assay kit. Go from an FCS file to a high-parameter analytic report in as little as 5 minutes. Obtain analysis results independent of user proficiency and manual gating bias. Maxpar Pathsetter is also flexible, allowing you to build a custom automated data analysis model if you want to add antibodies to the Maxpar Direct Immune Profiling Assay. Maxpar Pathsetter has an easy-to-use user interface. Multiple FCS files can be loaded and analyzed as a batch. You simply click Auto to have Pathsetter automatically run data cleanup and analyze your files. A few minutes later, the report will be ready for you to explore and export. In this slide, we see the Maxpar Pathsetter workflow. In step 1, Pathsetter generates an FCS file of single viable cell events. In step 2, population frequencies are calculated by probability state modeling. In step 3, the report is generated, which includes population frequencies as well as QC metrics and data plot displays. Let's discuss each of these steps in a bit more detail. In the first step, Maxpar Pathsetter's automated analysis, viable single cells are selected and a new FCS file is generated. The automated cleanup removes beads, debris, dead cells, and aggregates. The plots on the slide show data before and after the automated cleanup is performed. It's important to note that these plots show the results of the cleanup by showing specific plots before and after the selection is applied. These plots do not represent the way the algorithm actually performs the single viable cell selection. In the first row of plots, you'll see data before cleanup is applied. Dead cell debris and doublets are all present. These same plots, after the cleanup has been done, show removal of the dead cells, doublets, and debris. In the first two plots, you'll see live single cells, as indicated by the tight DNA peak, have been selected. In the last plot, which shows CD3 versus CD19, you'll see that the CD3, CD19 double positive cells are now gone, and as you know, these would be largely due to doublets in a peripheral blood sample. In step 2, Maxpar Pathsetter identifies populations and calculates frequencies. A B cell example is shown. In the model, 2 B cell populations will be identified, namely naive and memory B cells, using CD19, CD20, CD27, CD38, and IgD. The progression plot shows how these markers change between the naive and memory B cell populations. Traditional dot plot views of marker expression, colored according to the cell population, can also be viewed. In step 3, a report of results is automatically generated. The report summary identifies 37 immune populations and also reports out the number of cells per population, the percentage of total intact live cells, and the percentage of the parent cell population, as well as QC metrics. The report can be printed as a PDF or viewed in an Excel spreadsheet, as well as having the ability to customize the report to your needs. The high-dimensional Cen-se map allows the data generated by Maxpar Pathsetter model to be explored. Since the model and Cen-se analysis are fully integrated, the definitions for cell populations identified by the Pathsetter model are also used to identify these populations in the Cen-se map. This feature provides an important benefit over using a separate t-SNE plot. By using the Cen-se plot, you can also visualize new markers or populations defined by any drop-in antibodies added to the Maxpar Direct immune profiling assay panel. Now I will turn the presentation over to my colleague, Thiru Selvanathan, who will present data supporting the ability of the Maxpar Direct immune profiling assay and Pathsetter software to deliver consistent and reliable performance lot-to-lot, run-to-run, and site-to-site.
Thank you, Michelle. Here is a workflow for testing lot-to-lot performance of the assay and software to identify 37 immune populations. To do this, one technician stained a single whole blood donor in triplicate with 2 different lots of the dry antibody panel. All tubes were collected on a single Helios mass cytometer and analyzed with Pathsetter software. The percent of 35 immune cell populations identified by Pathsetter is displayed. The graphs display data for the combined 2 lots of antibody. Note that the data is divided up into 3 graphs according to population frequency. First for populations of greater than 10%, the second for populations between 1 and 10%, and the third for populations under 1%. As shown by the small interquartile range for each population, the variability of antibody lots is low for the combined data. Another test was performed to compare the performance of the dry 30 marker antibody panel to the same panel in liquid format. Whole blood and PBMC from a single donor were processed independently by 3 technicians. Each technician stained both cell types with the liquid and dry formats. All tubes were collected on a single Helios mass cytometer and analyzed with Pathsetter software. The liquid and dry versions of the assay were compared for their ability to identify 35 immune cell populations. As shown by the high R-squared values, validation of the dry panel on the y-axis against the equivalent liquid panel on the x-axis allowed identification and enumeration of the 35 populations equally well in both whole blood and PBMC. The repeatability of the assay was tested by staining and collecting data for both a whole blood and PBMC sample on a single day with replicate assay tubes from the same lot on a single Helios instrument. For the whole blood sample, a single technician stained 8 tubes. For PBMC, 3 technicians stained the same sample in triplicate. The top row of graphs show the results for the 8 individual assay tubes prepared from a single whole blood donor. The bottom row shows the results for the 9 individual assay tubes prepared for the PBMC sample. Note that the data is divided into 3 graphs for each sample type according to population frequency. The first for population with frequencies greater than 10%, the second for population with frequencies between 1 and 10%, and the third for those with population frequencies under 1%. Note how tight the distributions are for all populations 1% or greater. Next, we want to determine the level of agreement between manual gating and our automated results generated by Pathsetter. The manual gating was performed in CytoBank using a standard gating strategy for each population. Examples of the gating can be found in the CytoBank public experiment titled Fluidigm Maxpar Direct Immune Profiling Assay 201325 Gating Example. Here, we graphed the frequency results for some of the major cell populations found in whole blood and PBMC for 15 different donor samples as determined by manual gating on the x-axis and Maxpar Pathsetter on the y-axis. We performed demi-regression to compare the 2 different methods of analysis. For each population, the 95% confidence interval of the regression coefficient for each subset is indicated by the pink shading. The 95% confidence interval of the regression coefficient contained the value of 1, suggesting that there is no proportional difference between the 2 methods of analysis. A final and very important test was done to compare the reproducibility between sites from the same whole blood and PBMC samples stained in multiple replicates by a single technician at each site and collected on the site's Helios instrument. 3 sites were sent 1 whole blood sample and PBMC sample in week 1, and 3 additional sites were sent the same donor samples in week 2. All sites received the detailed protocols for sample preparation, an assay-specific acquisition template, and instructions for data acquisition on a Helios system. The PBMC sample at site 6, week 2, was not processed according to the Fluidigm protocol provided, and so that data was not used. All data was analyzed at each site using Maxpar Pathsetter software and following the provided instructions for use. Here are the plots for whole blood population frequencies from all 6 sites. Again, note that the data is divided up into 2 graphs according to population frequency, the first for populations of greater than 10%, and the second for populations between 1 and 10%. Do note, populations with frequencies greater than 5% have CVs lower than 20%. Here are the results from the PBMC samples, which also show low variability across all sites. Note that the data is divided up into 2 graphs according to population frequencies, first for populations greater than 10%, the second for populations between 1 and 10%. Populations with frequencies greater than 5% have CVs lower than 20%. Considering all the potential ways that variability can be introduced in such a study, we believe that this data is very impressive and compares extremely well to data generated by similar multi-site flow cytometry studies. Michelle, I'll turn things back over to you now.
Thank you, Thiru. In summary, we've introduced you to the Maxpar Direct Immune Profiling System components and shown its performance data. The system consists of a dry format 30-marker assay tube, including all necessary reagents for processing whole blood and PBMC samples, the Helios mass cytometer for sample collection, and Maxpar Pathsetter software, which identifies 37 immune cell populations automatically and produces a detailed report of results in as little as 5 minutes. The assay can also be customized by addition of 7 or more markers labeled with isotopes corresponding to open channels on the Helios instrument, and a custom data analysis pipeline can be created in Maxpar Pathsetter. We've also demonstrated the reliable performance of this system lot to lot and run to run. In addition, the results of multi-site testing show that consistent performance of the system as a whole can be obtained even when different technicians at a variety of sites process samples and collect and analyze data independently. We hope you're convinced that the Maxpar Direct Immune Profiling Assay sets a new standard in immune profiling. 30 canonical immune cell markers that in concert identify at least 37 immune cell populations, all in a single tube of sample. The combination of a pre-optimized, easy-to-use, high-dimensional immune cell panel for analysis by mass cytometry with an automated software solution provides the most powerful and reliable way to analyze your precious PBMC and whole blood samples time after time. The flyer for the system can be downloaded from the Wiley webinar site. You can also request more information or a seminar at your institution at fluidigm.com/immuneprofile. Thank you for attending the webinar.
Thank you very much for that presentation today. Some of the questions that we've had come in are there different protocols for staining PBMC and whole blood? Does one take much longer than the other?
So this is Thiru. There are 2 different protocols for the PBMCs versus whole blood. PBMCs can go directly to antibody staining using our dried down antibody pellet. Whole blood we recommend being collected in heparinized tubes, and prior to staining we recommend that you do an additional heparin blocking to reduce the amount of non-specific binding. And then you would continue on to do antibody staining on these heparin treated whole blood samples. And then following antibody staining, we recommend doing a red blood cell lysis using cal-lyse from Thermo Fisher. And this takes approximately an additional 40 minutes to do the lysis and the washes after.
It's kind of a follow-up to that. Does the protocol work well with both frozen and fresh PBMC samples that could be used with a kit, or are there any kind of differences?
We have tested the kit on frozen PBMCs, and it works pretty well. We have done some limited testing with fresh PBMCs, and I'd have to get back to you on that.
Does the whole blood have to be collected in a certain kind of tube?
As mentioned earlier, we tested and developed the assay on heparin, used in heparinized tubes. So we recommend collecting your whole blood in sodium heparinized tubes and using that.
Okay. Are there any things you can do if your sample has stained poorly or has a low viability to it?
In terms of the data analysis tools for Pathsetter, it's really great because it has 2 modes where it first looks at the quality of the sample or of the data that you collected, and during this step, it will identify viable cells as well as any debris and aggregates. So it'll give you an indication of how the viability of your sample, and then following that, it will do the immune profiling analysis on those viable cells, and so it will give you an idea of how well the samples and the staining worked. In terms of if you do have a poorly stained sample or have low viability, we can work with the field application scientists to troubleshoot that.
Okay. So what if an individual is looking to do something like this, but they don't have a mass cytometer at their institution? Are they able to ship samples of PBMC or whole blood to a collaborator that has that or a core facility and have it run there?
Yes, if you have a collaborator at the site with the mass cytometer, and this kit has been developed for the Helios mass cytometer or the CyTOF 2 to Helios upgraded mass cytometers, you can ship them your frozen PBMCs, or you can ship whole blood that has been collected within 48 hours, and they can do the staining on site and do the acquisition and analysis there.
In terms of whole blood analysis, can you give any more information about the blood collection tube and the time between blood collection and staining?
In terms of the blood collection tube, we recommend using sodium heparinized tubes. In terms of the staining following the draw, we recommend you do that within 48 hours to get a good quality staining.
When you're processing things, how much consideration needs to be made when assigning channels to the additional 7 markers?
This is Michelle. I'll take that one. What I would recommend is for anyone who wants to optimize the panel and assign channels to additional markers, that they contact their field application specialist from Fluidigm. The field application specialist can help them to make the best metal choices to get the most optimal panel possible. It's certainly not a very difficult thing to do, but it's good to have an expert to give you some advice in the process.
In terms of the analysis software, to be able to get your results in 5 minutes, what kind of computer capacity do you need for this? Do you do like a supercomputer cluster or just a resident computer in someone's lab or clinical setting?
Nope.
Go ahead, dear. Sorry.
So this doesn't require a supercomputer. This would just require what most laptop specs have. So this would require a CPU of 3.4 gigahertz or greater with 8 gigs of RAM. And we recommend a free disk space of at least 750 megabytes.
Okay. In terms of the data that's generated, will you be able to see 2D plots of the data, or do you just get a table of that and then that you then have to plot yourself? So you will get both the population frequency and event counts, as well as getting 2D plots of the markers, and you can within Pathsetter, you can actually create bivariate plots as well. What is the appropriate white blood cell count to use for whole blood staining? We don't have a definite recommendation in terms of a white blood cell count prior to staining. In the hundreds of samples that we've tested, we found that the assay worked really well. Okay. How would you recommend optimizing drop-in antibodies?
So, remember that is right to be direction on that.
Sorry, go ahead.
Yeah, so I think Michelle is said to reach out to your field applications specialist in order to get help with with dropping in or adding antibodies to the 7 open channels. What you at least keep in mind is the same panel design considerations as you would when you're creating a new panel. In this case, you're creating with a base panel, and so you're looking into how expression of the antigen as well on per cell and on which cell types.
Could you use a sample collection tube that stabilizes the cell, such as a STREC?
So I'm not familiar with the STREC, but if this is similar to fixing the cells before staining, we don't currently recommend that. We haven't validated the assay to that because following fixation, you will have some markers that are diminished in their staining intensity.
Okay. Do you have recommended storage conditions for whole blood prior for use with this system?
In terms of storage conditions, we would say collect it in sodium heparinized tubes, and you can store that at 4 degrees until you're ready to stain within 48 hours.
Okay. So does this protocol include a fixation step, and if it does, how long can the stained specimen sit before acquisition?
So yeah, both whole blood and PBMC staining require a fixation step prior to the last iridium staining. So in mass cytometry, iridium staining is used to identify nucleic acid positive events. So you're looking for cells. Prior to that, we would do a fresh fixative staining for 10 minutes. And following iridium staining, the sample is good for 48 hours before acquisition.
So some of the other questions that have been coming in, is the instrument CFR21 Part 11 compatible for GMP work?
No, the mass cytometer Helios is not, CFR21 Part 11 compliant. Pathsetter does have a logging feature where it will log changes made to the analysis of a particular file. But the analysis tool and the instrument are not CFR 21 Part 11 compliant.
Okay. Do any locations provide the services to check blood samples? And do you have an estimate about how much this would cost?
I'll take that one. Hopefully my line is a little clearer now. Again, the field application specialist can help you to get in touch with core labs to discuss full service for this panel. I can't speak to what the cost would be because that will probably vary between cores, but there are cores that take outside samples. We can put you in touch for specific information on this panel and help to get you some pilot data.
And do you actually have any recommended core facilities that are providing services such as this?
There are a number of cores. I think it would be better to speak specifically with the customer to see what their actual needs are, and then put them directly in touch with the core.
All right, well, I believe we've reached the end of our question and answer time for today. I would like to thank Dr. Poulin and Dr. Selvanathan for taking the time to speak on today's topic. This presentation was sponsored by Fluidigm in partnership with Wiley's Current Protocols. This webinar will be available for on-demand viewing at the Current Protocols website in the webinar section. Thank you for attending.